I think the important thing last time was that the winemaker or the wine microbiologist has to decide, first of all, whether the problem with the wine that he's going to look at, examine, whether it's microbiological or non-microbiological. And once he's made that decision and it's microbiological, then he's in business. And then he has to decide whether, from morphology under the microscope, whether it's most likely a yeast or a bacteria, because he needs to use differential plating techniques depending upon which kind of organism it is. And then he hopes that the organisms are still viable, that the wine isn't so old, or that the spoilage hasn't been for such a long time past that the organisms have already died. Then he wants to get the organism out. He wants to cultivate it, I should say, and to purify it. This is important. He might be doing tests on the wrong organism, getting the wrong answers. And he wants to identify it. Now, we said that that probably isn't the first thing on his mind, because mainly it takes a long time to do this. But it is important that he do identify it. The quicker the better, because he can get some insight of how other people have handled the problem if he knows what the organism is. And so with this as my idea of what the take-home lesson is, I wonder if we could have a little discussion, if anybody has any questions on what we talked about last time before we go further. Don't want to have any confusion at the beginning. Yeah?
Well, this is sort of dumb. How can you be sure that the organism that he's isolated is the one that's causing the spoilage?
Well, that's not a dumb question. It's a very good question, and sometimes perhaps you're getting a different one out. In my experience, generally, you see the wine, if it's a microbiological problem, there's lots of organisms there. There's a high concentration of usually one kind of organism, maybe two. And it just seems, and the problem is very recent. The guy's got spoilage that probably just recently happened. So the chances are that what you get out is the one that's causing the problem. Or the one that you get out is the same one you saw under the microscope. That's another question. So you have to always keep going back and examining the wine to see if that's the organism that you've isolated. You might be isolating something that grows much better, and it's only there in a very small amount. There is one disturbing aspect of this. Lüthi, a famous Swiss microbiologist, enologist, has said in a paper that we'll read perhaps, that he's never had a case where he's isolated an organism and put it back into a wine and got the same kind of spoilage. This is like Koch's postulates that you should be able to do that if that's the organism. He says he's never been able to. We've been able to get things happen, but I think that what he's saying is that there is a problem of the conditions of the wine may have a lot to do with the kind of spoilage, not just the organism, and you do have to be aware that you may not be isolating the organism that caused the spoilage. Go ahead.
I have another question.
Yeah.
But what advantage is it really to the winemaker to isolate the organism? I mean, there are very few things you can do in case of spoilage. I think he wants to be able to do those few things. His job isn't so difficult, that's true. He'll want to look at its heat sensitivity, its SO2 sensitivity. And also I think he wants to assure himself that is the problem, that it is microbiological. Yeah.
What do you do if the cells aren't viable?
Nothing you can do. Yeah. I suggested about ways that if the viability is very low, how you might increase your chances of getting a viable one. But as I said, in no case I know that made any difference. Usually, so you can take a case where wine has been made here and say sent to someplace else, eastern United States, maybe to the Panama Canal, or maybe even into Europe, or some distant place, and the spoilage happened early, and then you would have wine that had been spoiled by a microbe, and it was very long time ago, and you might not be able to solve your problem. But generally speaking, that's not what happens. So it's happening in the winery, or the bottle just brought back by a customer. I hope that doesn't happen. But it's usually a fresh situation. And no, I can't. I've had, even here though, I've had some organisms that, well, they weren't really spoilage problems. I mean, it wasn't really an economic thing, but somebody brought it to me and says, something's wrong with this wine. It seems to be microbial, and I wasn't able to get anything out of it. So, I have one more question here, John.
Is there any chance that spoilage might only occur if you have two organisms?
You mean rather than one?
Right.
Yes, I don't know of any documented case, but what they call the synergism effect, where one organism helps another. There are cases where, not for spoilage, but you have two lactic acid bacteria growing. One requires, say, vitamin B1, and another requires another vitamin. And each one makes excess of the other one. So both organisms will grow together where they might not grow separately. Okay, one more question. We'll go on. If there's another one. Yeah.
Do you ever get one organism that's spoiling the wine replaced by another one, so as the alcohol content rises?
Again, I think that's a possible. As alcohol rises, you mean during the alcoholic fermentation?
More tolerant of alcohol, higher alcohol content.
Well, usually these spoilages don't happen. They happen in the storage of the wine after the alcoholic fermentation. So the only way the alcohol would change is if it had been fortified. But I could imagine a case where you had a spoiled wine, and then you fortified it, and you got Lactobacillus trichoides. I don't know of any case it really happened. And I guess another take-home lesson is that it's not really, generally speaking, not a big problem. We don't have many cases of wine spoilage. It's one case, as I said, when we just have so much wine in the winery, we can't take care of the situation. In that case, it's not a problem for the microbiologist because everybody knows what to do, how to solve that. And the other case was with this, the Brettanomyces yeast, which is kind of a new situation. I went very hurriedly and very vaguely over several yeasts that are spoilage yeasts. And the reason I had to do that was there just isn't a whole lot I can say about them. But when I came to Brettanomyces, I had quite a bit to say, and I did. Yeah.
Has there been an instance of a winery that makes core sherry as well as table wine in the same setup?
Yeah.
Where core sherry yeast contaminate the table wine?
No, I don't think so because it needs air. It needs air, the anaerobic. Probably floor yeast will carry out the alcoholic fermentation, though. I guess we're not using one of those for our experiment. Well, let me just finish up one thing about Brettanomyces that I didn't say was that I said, what does it live on? And it seems to, as an energy source, seems to use alcohol, either forming acetic acid. This is one of the things, one of the hallmarks of it is you get an increased VA, or else taking it to CO2 and water. There is another possibility, and we haven't checked this out, but that's what this whole course is, what part of this course is for you to see what new is going on, and it may be important. Somebody reported a wine, a white wine, that had not undergone malolactic fermentation yet, and at the top, the malic acid was gone. At the bottom, the malic acid was present. And they suggested that this is a very interesting malolactic bacteria, which seemed to be more than microaerophilic, perhaps, and would only grow up here where the high redox potential is. Well, we examined this wine, and there was some bacteria there, but there was also Brettanomyces. And I'm just wondering if in the presence of air or a high redox potential, that Brettanomyces can also oxidize malic acid, say to ethanol and CO2. This is speculation, but it could be. Most cases of this report has been most cases, at least in California, of the Brettanomyces infection has been in wines which have already undergone malolactic fermentation. So there wasn't any malic acid there. But this is something kind of interesting, and we want to look into that. Okay, where's my notes here? Oh, there was a question came up about the media I was talking about last time. I went over a little bit rapidly about adding fructose and adding malic acid to the rugosa media. Or in lactose too. Ordinarily, we make up this medium with just glucose, because that seems to be a good energy source for everything. Historically, or I should say other people, dairy people, other lactic acid bacterial people, always add lactose also, because they're looking at bacteria which grow from animal sources or from dairy sources where there is lactose, and they can use that as an energy source. We discontinued adding it because none of our organisms ever use lactose. But we do add fructose because it's been reported, and we know from our own experience, that fructose stimulates the growth, early stages of growth of some lactic acid bacteria. And we add malic acid too because we know that malic acid stimulates the growth rate of some malolactic bacteria, at least some of them, maybe all of them, but at least some of them. So that's why we've added these things to the medium. got some handouts for us. This first one I'm passing around is a bibliography of wine texts and books. Yeah, maybe we better take a few off the top there. I guess it does very well. Might as well pass the other things out at the same time. Here's a list of some yeast taxonomies. Maybe you can pass them each way behind you. And here is a taxonomy written by me. Handle it carefully. And the last is the experiment for Thursday. I'll let you get those before I go further. Okay, the Abridged Wine Microbiology Bibliography is not necessarily intended for this course. It's just something that I have for people to write in from time to time. But we will try to have most of these books on the reserve shelf in the Winkler Library. And again, that can be checked out for short periods of time for Xeroxing if you want to Xerox any part. In addition, I have some like this listed as reprints. And for example, on the second part of that page, back, chapters and texts, about midway through it, it says malolactic fermentation, R.E. Kunkee. Put a Roman numeral 1 by the side of that, and that's an extra copy of this that you can take easier to handle for Xeroxing. And that'll be, and you can just sign your name on here and give it to Mrs. Hendricks and then bring it back in 5, 10, 15 minutes, whatever it takes you to Xerox it. Above that is yeast and winemaking. Do you see that? By Kunkee and Amarine. Make that Roman numeral 5. There's another one here. Oh. Maybe I can collect some from one side. And are there any extras? People got extras. Jed? Hand me? No, hand me. Okay. Well, let me get all the extras together. There must have been. Okay. Here's the bibliography ones. Who needs that? Okay. All right.
Should these be in the Winkler Library?
Yeah, they will be on a reserve bookshelf for 217. Okay. All right. Any more extra things? Well, we'll wait till we come to it. Yeah. Was it next time on? Okay. Okay. So back to this bibliography. Okay. The yeast and winemaking on page 2 is Roman numeral 5. Malolactic fermentation is Roman numeral 1. It's just these reprints that we have separately that can be. Does anybody need the it's on the back of it. This bibliography has page 1 and page 2. It's on both sides. It's one sheet of paper. So you turn it over. Okay. On page 1, where it says identification, bacteria, yeasts, and J. Lauder, the yeasts, that's Roman numeral 4. Not the whole thing, but a key to the genera. We'll talk about that in a minute, I hope. Okay, it says identification, bacteria, yeasts, J. Lauder. Roman numeral 4, part. It's not the whole book. This is the whole book. And also, I've got a series of 4 reprints on Brettanomyces, which you could list as Roman numeral 2. Okay. It's not on there. That's for your own information. It's not going to, none of it's assigned on the Britannomyces. If you thought it might be information that some of you may want to have available. Okay, so I think you understand how I hope this works out well. I've tried it different each year. And
I don't know how to relate to these Roman numerals. What are they?
well, where it says malolactic fermentation? Yeah. Well, that book, Advances in Applied Microbiology, will be on the shelf. And also the reprint itself, a Xerox reprint, which will just be easily identified as Roman numeral 1, but also it has the citation in it too.
Okay, when the yeast wine
Okay.
made by yourself and Dr.
Yeah.
Emery, what Roman numeral is that?
That's 4. Not 5. And the book itself will be on the shelf. Number 3, well, that's a little complicated, and I'll take that up in a minute when we get to that. It's listed on one of the other sheets. Okay. Well, let's get on to, now we want to talk about differentiation of some wine yeast strains. And I can give you some supplementary reading for that. Roman numeral 5. See how easy that is? This is not assigned, but it may help you. Those pages. Again, we're kind of putting the cart before the horse. We're going to talk about yeast strains, different wine yeast strains, when we haven't even talked about genus and species yet. But we want to get this aspect out of the way because we want to do an experiment on Thursday based on some information about wine yeast strains. This is probably the most interesting part anyway, because this is, I think, what we're all like to think about is how different wine yeast strains may affect the rate, the fermentation, the flavor of the wine, let's say. And there's interesting history about this, which I won't go into too much detail, but something you should know something about. And for that, there's a signed reading, page 6 to 9 here, Roman numeral 5. This is a little bit of history of the selection of wine yeast strains. Starting from Pasteur on. Now, I don't expect you to memorize dates or names, except a few names. You should know who they are. Pasteur, Hansen, Müller-Thurgau. The Great Brothers. Well, Pasteur, as you know, started it all as far as microbiology goes and gave us a lot of information on what happens during alcoholic fermentation. And then he did get involved in wine spoilage, and there is a famous book I meant to bring in, but one of the students had it checked out. I'll show it to you next time. It's a really, very interesting book, lots of nice drawings. He didn't have any access to single-cell cultures, you know, single pure strains, in the sense that we would have, because solid media had not been discovered in his time while he was doing his microbiology. Yet he didn't do a lot about wine yeast strains. Hansen did this for beer, for the beer people. He selected a lot of beer yeasts, and you can see that it was much more important for the beer people because the beer, the wort, or the malt is sterilized, is boiled, and so you have to pitch, you have to add some source of yeast. And so it became once they knew about microbes, it became very interesting to see which strains would be best. Müller-Thurgau, who is not a grape but a man, which the grape is named after, followed in some of the ideas of Hansen, doing this with wine. But it never really got off the ground. We really don't have, in most places of the world, strains that are used specifically for certain kinds of fermentations. Now, that's not true in new areas where wine is being made, like California, Australia, and South Africa, where we almost always add a starter culture. At first, there were great claims made for these, that if you used a yeast from Burgundy, you could make Burgundy wine, no matter what the grapes were like. Well, this just doesn't hold up. How important this yeast strain is, we'll decide at the end of the quarter with our own experiments. When we talk about the effects of yeast on winemaking, we can look at it two ways. One, we can look at pure cultures, which is what we're going to look at it right now. Or there's the other possibility is using mixed cultures, or trying to obtain what would be like the spontaneous fermentation in Europe, where you might have a succession of yeast causing the fermentation. Now, both of these could have an effect on grape on the wine quality. We're going to stick right now with using a pure culture, one culture, and see its effect, or the differences between different cultures. Well, let's, I think you're sophisticated enough to know what kinds of things we'd have to look for if we wanted to differentiate between different yeast strains. Now remember, this is not like we do with genus and species, where it's yes or no. This organism will ferment glucose or it won't ferment glucose, or it makes spores, sporulates or it doesn't sporulate. We're talking now about subtle differences, which aren't yes or no. They're just the differences in quantitative, quantitative? Yeah, quantitative differences, where one fermentation rate might be faster than another. Let's see, we have a little trouble with chalk here. Margaret, think you could run into, oh, here, we're okay, we're fine. Let's list some of the things that we might want to look at to see if we could tell one yeast strain from another. Anybody want to, what?
Rate.
Rate, okay, what rate?
Rate of the fermentation.
Okay, we talk about the fermentation rate. I looked at you and I says, what rate? What else could I be talking about?
Growth rate.
Yeah, the growth rate. You might not realize that there is a difference there. That the yeasts are put in a low inoculum and then they grow and ferment. And so the growth rate could be different. I mean, one that fermented fast might grow slow. Anything else?
Alcohol tolerance.
Very good. Alcohol tolerance. Anything else? SO2 tolerance. Yeah. Okay. Could say both hot and cold. Heat and cold tolerance.
Whether it ferments dry or...
Yeah, well, I think that would be alcohol tolerance. Probably. There might be some, you could imagine a case where a highly tolerant organism didn't ferment to dryness because of some nutritional defect, but I think it would be rare. Anything else you might want to say? You mean pH tolerance? Yeah. Practically not. They're about the same. But there is something about the amount of what the pH of the wine is at the end, the final pH, and titratable acidity. This can vary from strain to strain. That means a yeast can make acids, particularly say lactic acid, or it can utilize some acids like malic acid, and they all do this a little bit. The famous one, there's the Schizosacchaромyces, which will use up almost all the malic acid under certain conditions, and it'll really change the pH. It's a way of deacidifying very highly acid grape juices. Anything else?
nutrients.
This is really difficult. There's a whole history on trying to classify yeast strains by the nutrition, and you look at that literature and it's just a mess. They really can't. It's very hard to do. One thing, they all seem to be biotin requiring, but this is very hard to show because biotin's everywhere. It's very difficult to get a biotin deficient situation. So I would say we wouldn't talk. We'd leave pH tolerance out, and we'd leave nutrition out.
Alcohol and fermentation?
Yes, that is more or less so characteristic of the material. No, the material being fermented. We could talk about film or top or bottom. Let's put it down anyway. Anything else?
How about release of H2 gas?
Oh yeah. Let's say about end products, and list that as one of them. Anything else? Yeah.
Flocculating characteristics?
Yeah, that's very good. Flocculating. Not too easy to measure, but it's certainly a great difference between some strains. Anything else? There's a few more. Talk about end products. Can you think of anything else?
CO2.
That's going to be part of the species differentiation, I think. Whether they're going to ferment sugar or not, unless you mean about the speed of fermentation. It's not very. Again, it's this It's not very good marker. Efficiency, conversion of sugar to alcohol. I'll put that in iffy. It's about the same for all of them, but we could put it down too. No, I think that's going to be the same from strain to strain. Yeah.
How about tolerance of CO2? Tolerance of
Well, that's interesting. I don't know what it is between strains. I don't know if you're aware that yeasts are very interesting in that they can't stand about more than 5 atmospheres of CO2 pressure. Bacteria can do this very nicely, but yeasts don't. I don't think there's much difference from strain to strain, but I don't know if it's been tested. We did some work on that. I thought it was kind of interesting, but not from strain to strain. I'm going to leave that out. Anything else? What about end products? What else? Oh, yeah, we put diacetyl. If I can spell it. And if you can read it afterwards. What else? Another one that goes along with this, maybe. Fusel oil, yeah. And what goes along with this, maybe? SO2. Yeast make SO2. We'll see about that. Anything else we might want to talk about? Let's see. Yeah?
How about just pure lab things instead of this like staining or generally base coats?
No, I don't think. I think they'd be the same. That's usually species differentiation or even genera.
No, well, there are some stains now.
But
Strain stains.
strain stains. Well, I don't know about them. You have to talk to me about. Henry?
Lewis was talking about them for beer.
Oh, yeah?
They have, yeah.
Oh, maybe you're talking about antibiotics and fluorescent antibodies for different strains. Yeah, that is a way for detecting wild yeasts in a brewery. That's right. But I don't think that's for strains, but it might be.
Yeah, he said it was for Cerevisiae.
Yeah, but does it differentiate one strain from another? I don't think so. Differentiated wild yeast from Cerevisiae, I think.
That's what these.
Yeah. Well, we'll talk about it later. Yeah. Maybe somebody don't know about that. Okay. Any more? Okay, you can put that down too. That probably is not going to be much different, though. One other thing I had, I think. I have a question mark about foam. This came up recently in some discussions that this could be important. If you could get yeast strains with low amounts of foam, you might have a better chance of filling up your tanks, even though antifoams are being used. I put a question mark on that one. Another thing on this fermentation rate, we can divide this into a few things. We can talk about the overall rate, and usually that's measured that people talk about the time from 20-degree Brix to 0, which in a sense isn't overall, but it's a kind of a standard measurement, which may not be the same as the maximum rate. What I mean is that one strain may have a higher maximum rate, but this overall rate might be slower because it slowed down toward the end when the alcohol got too high. And another thing is the lag time, which I guess I shouldn't really put that in because that's related to the growth rate. Well, there's one other thing, important thing left out, most important of all. The taste of the wine. So let's put that down here too. So what we're going to do on Thursday is set up some fermentations for fermentation rate and for making wine for some of the end product analysis and for tasting. And that's handouts to be given. I hope you read that before. There's not too much to do on Thursday, but there's a lot to do next week where you have to kind of organize yourself into groups to be able to do the Brix readings. Well, let's go on now then to yeast identification itself. Let's say that we have isolated the strain, like the organism, the colony, the strain, as we will do in the next 2 or 3 weeks. And we want to store it, first of all. We don't want to lose it. We want to make a master slant of it so that we can go back to that. And I think you all know this, and we'll have slants in the lab where you have the auger, the cap. The auger's like this. For yeast, you just streak this out here. It grows. And then you can store this for long periods of time. The biggest problem is that the auger dries out. So to prevent the auger from drying out, you can wrap this with parafilm, and store it in the refrigerator for a year or longer, and it'll transfer very nicely. There's some growth that's going on here at this low temperature, but not a whole lot. Another thing you can do is cover this with sterile mineral oil. And this, if the whole thing is covered, then this prevents any drying. Does anybody know how to sterilize mineral oil?
Put it through acid.
That probably could do that. That's not the way we do it. You can see there are some problems. You don't want to really put it in an oven. You might hit the kindling temperature, whatever that is. Well, you can autoclave it, but you can't just autoclave it because it's dry, and dry things don't sterilize at autoclaved temperature. So you have to add some water to it. And we do it like tindelization. We autoclave it for an hour on 5 different days, 5 successive days, an hour each time. And it's never had any problems since then, doing it that way. Okay. The only exception about this where you have to worry about is with Brettanomyces or Decara. You have to have, say, 2% calcium carbonate in the medium and suspend this just before it solidifies, so the calcium carbonate is throughout this, so it can neutralize the acid that's being formed. And actually, you'll see a little bit of clearing around where the yeasts are. That's not a diagnosis of Brettanomyces. Well, if you get it, it would be. But just because it doesn't make that much acid, so you get clearing, wouldn't mean that it is not Brettanomyces. There are other ways to diagnose Brettanomyces. Well, we've already mentioned why we think we ought to identify the organism and got some rebuttal on that. One of the reasons is because I think you should. No, as I say, you probably, if you can identify it, you have a head start on how to control it because somebody else has probably had the same problem. I think before we go further, we ought to define what a yeast is. Does anybody have a nice, handy definition of a yeast? Okay, you have one for fungus, then maybe we can go from there. Well, let's start a little further. I think it's good to take 2 minutes and talk about where the protist kingdom is, where these organisms are in the protist kingdom, because we're going to be talking about wine yeasts and wine bacteria and how do they fit in with other living organisms. So if we talk about the protista, what are the 2 main divisions we have, these microbes? We mentioned them the other day. You have the eukaryotes and the prokaryotes. Prokaryotes. And these, as we say, had a true nucleus, and these don't have any nucleus, a true nucleus. So what 3 main divisions do we have of the eukaryotes? Anybody remember? One was mentioned, fungus. What else?
Lime mold.
Well, that goes in there, I think. Yeah. Or in bacteria. What? Algae. And what else? And protozoa. Well, if we're going to, first of all, where are the yeasts? The yeasts are a type of fungus. Yeasts are defined as organisms, a fungus that spend, or the predominant part of their life cycle is in a single cell form. You get that? So yeasts are just a special kind of fungus that predominant form of its life cycle is in single cell form. It's not a taxon in that sense. It's not a bona fide classification of a kind of organism. I think before we go further, perhaps we ought to see if we can distinguish between these 3. And what's the distinguishing characteristic of anyone? Yeah.
Well, algae.
Okay.
Photosynthesis.
Right. Photosynthetic. What? Well, not necessarily, but I think you got the right idea. It has flexible walls that it can do things with, eat with. Cell wall. So this leaves the fungus non-photosynthetic with non-flexible walls. And then, so that's where the fungus is. Then the yeast is a single-cell fungus. But while we're here, let's talk about the prokaryotes. You want to name what they are? Bacteria. Yeah. And what else? Is there any other division? Blue-green algae. Rickettsia, that's getting down a little smaller. We'll leave that out. Blue-green algae. Blue-green, yeah. And what's the difference between these two? Photosynthetic, yeah. So the things we have to worry about from wine then are these, from wine itself. So we're all set now. We've got the yeast. We can go ahead and start classifying the yeast, right? Now, what do we need? We need a good taxonomy. And there are several. Let's get the sheet out that says yeast taxonomies. Does everybody have one of those? Okay, let me get some. Anybody didn't get one? By the way, did everybody get the number 3 handout, the experiment, wine yeast fermentations? Okay. Okay. There's I've listed here 3 yeast taxonomies. The first one was the classic one by Lauder and Gregor von Rhee, and this was published in 1952 with a new edition. I shouldn't say new edition, a new reprint of it in 1967. Now, it's out of date as far as the taxonomy itself goes. The new taxonomy includes new genera and some new names and a new way of classification. Yet this does have importance because it does have a lot of historical material in it for description of different species and genera. I'm glad we have it. I don't know if I would say we should buy it if we didn't have it, but I'm glad we do have it. One thing I should say, that the way that you should be aware of, because a lot of people don't know about the new taxonomy yet, is the way that these authors classified the organisms in this taxonomy, and it's listed here. They classified them by their mode of sexual reproduction, if any. If they formed ascospores or ballistospores or no spores at all. This corresponds to the 3 main groups of fungi, Ascomycetes, Basidiomycetes, and the fungi imperfecti. So then they had these yeast families based on these 3, which was a sensible way to do it. The only problem is that meant the first thing you had to do was to find out whether there were spore-forming organisms or not. And this was often the most difficult thing to do of all. So I think we all very much welcome this new taxonomy, big as it is, big and expensive as it is. It's the separation of the genera is somewhat simplified. There are only about 30 genera, so we don't have too much to do. And of those 30, only about 15 are ones that we'd be interested in. By the way, in reprint number 5, in the back of that reprint is an appendix where we have listed, as far as we know, all the yeast names that have been associated with wines, wineries, grapes. I guess that's about it. And so there's about 15 genera that we have to know something about. One of the disadvantages of this, besides it being hard to handle, is that she doesn't use exactly always the dichotomous key. Now, a taxonomy is supposed to be a dichotomous key, where you have a red, say not red, and then you look down that, and maybe there's 2 other things. There's square. There are triangular yeast. Square, not square. Well, in this one of Lauder's, there are some cases where she'll have 3 different choices, and you have to do that. Then there's some cases where there's redundancy. You end up with 2 or 3 paths that you could go to get the answer, or you end up with not 2 or 3 answers for the same organism, but you could find the organism in 2 or 3 different places. This will be clear in a minute. I'll show you how this works. So this is a little bit of a problem. The last one there is down. This, for somebody who wanted to know it, this is Roman numeral number 3. this is a simplified scheme designed mainly for wine and beer people for identification of yeast. We've used it and it works, but I think it's not a very good teaching method, and I think frankly the one that we've developed for this class is probably a little better anyway, but it's one that I want you, not that I want you to be familiar with. That sounds like an assignment. I don't mean that. I mean that you may want to familiarize yourself with, because you may want to use it sometime. Okay. Let's go then to the taxonomy that we're going to use. And that's this one. It says wine-related yeast genera accepted by Lauder, 1970s. Everybody have one of those? And I think the best way to both to explain the key and to explain what the tests are that we need to do is to just go through the key step by step. Now, that's an important statement at the top. Wine-related yeast genera accepted by Lauder. By accepted, I mean the genus names. There are lots of genus names that are used that are obsolete, but she has about 30 of them in here that she accepts. And of those 30, we have taken about 15. These are the ones that are important in wine. And I noticed at the top there I say it's taken from yeasts without permission. I don't know what she'd say if she knew that I had done this, but it's done now. In the parentheses, don't tell. In the parentheses are listed for your edification the other genera that are not really wine-related, but where they fit into this simplified key. Well, by simplified, what I've done is just excluded those organisms that aren't wine-related and just so compacted the key itself. All right, let's begin. The first choice, I want you to understand how a dichotomous key works. And so if you don't know what I'm getting at, come and see me afterwards. And we're going, one of the questions in one of the quizzes or the midterms would be to take a page of Lauder's key, whether it's wine-related or not, and I'll give you some definitions, or I should say some descriptions, and I want you from that key to key out the organism. That's just a test to see that you understand how a dichotomous key works. Okay, we have a choice, the first one, not of two things, but of three things. And this is by looking at the morphology of the organism, the microscopic morphology, seeing how the shape of the organism and seeing how it divides. Hopefully it's alive and is dividing. And we have the three choices of fission, or bipolar budding, or multilateral budding. And just to remind you about that. By the way, you notice we didn't say anything about morphology on this list. Neither colony morphology nor microscopic morphology. It's not good enough to give us any information. So fission yeast would be ones that divide by making a bar right in the middle. And they're very, very characteristic and a little different from bacteria. These split open, they look like this when it comes apart, kind of hanging on there and then finally breaking away like this. Now, bipolar budding would be where you'd have yeast. Often they talk about apiculate shape, but the budding always occurs at one end or the other end. And actually the bud scars from these things are concentric. Here, people have seen them on scanning electron microscope, micrographs. But multilateral budding, say you have yeast like this, it could bud out here, getting bigger and bigger, or it can bud anywhere along the side. What happens in this case, the cell can take about 30 buds altogether. It forms a bud scar, and no bud can come off there again. And when the yeast has at the maximum of about 30 bud scars, then it can't divide anymore, can't bud anymore. So we have these three choices of fission, bipolar budding, or multilateral budding. And right away, if it's fission, then, and it's from wine, or it's related to wine, then we can classify it as Schizosaccharomyces or Schizosaccharomyces, however you want to pronounce it. Is that part clear so far? We've already identified one genus. Well, what do you do when you get to this? Then you look up in Lauder under Saccharomyces, and she gives a description of the organism and then a key for the species. So right now we're only going to worry ourselves about the genus, the genera. Oh, say, okay, for bipolar budding now, we have another possibility, either no spores or spores found. Now this is difficult sometimes to be able to detect spore formation. And the way you do this, first of all, you have to kind of prepare the organism for this, and usually you like to grow it on a very enriched medium, say malt, for a while, on a solid malt. And then transfer this to special sporulating medium. And there are all kinds of sporulating medium, and different ones are for different organisms. And we have to, if you don't know what the organism is, you probably have to try all of them. There are things like V8 juice medium. It has V8 juice in it. Or they're just pieces of vegetable themselves, autoclaved celery or carrots. Or sometimes gypsum blocks that have been sterilized. You just streak out on them. Or some very deficient medium which has just a little bit of glucose and has a high concentration of acetate. As I say, there are 4 or 5 standard ones, and we'll use them all. And what you do then is streak out onto this spore media, and then after a few days, over a period of maybe a month, you examine this under the microscope and look for spores. Now, spores are pretty easy to find if you know what you're looking for, and we'll have some definite ones for you and some slides to show you what they look like. It's not like the spores on a bacillus, where they'll be round things at, say, for example, at the end like this. What happens here with yeast, the yeast is going to deform and form an ascus, and the shape of it's going to be different in the first place. And let's say for Saccharomyces then, say this is the vegetative cell, about like that. Then for the sporulating, I'm going to draw the spores first because it'll be easier. There might be 4 round oval spores in there, and then the cell wall will be kind of conform to that. Or maybe there'll only be 3 or something like that. Question?
Would you mind giving us your theory on why yeast revert to, I won't say revert, or go into a sexual cycle as opposed to just the asexual budding?
Oh.
I mean, why is it?
Well, I think that geneticists always think that this, they call this the perfect form, that they all did this, but somehow they've lost this ability. So why did they all do this before they lose it? It's for survival of the fittest. It's a good way to mix up genes, and so you're getting new genetic material all the time, and you're getting a better chance for the organism to survive. So I think that's just evolution, that you would have the sexual cycle. These spores, by the way, are not like the bacterial spores that are sensitive to heat. I mean, tolerant to heat. They might be slightly, but nothing like bacteria. They're a sexual phase. This occurs after meiosis, and each of these then has 1 N set of chromosomes rather than 2 N. Yeah.
But you always have to put the, what I was leading up to was, I understand there are some ways to use just like heat shocking or something like that rather than changing the media. But aren't you always putting them in some kind of stress situation? I mean, if they're just floating along, they're not producible.
Yeah, that's generally, you do try to put them in a kind of a stress situation. I said with low glucose and high acetate, and I said that you grow them well on a good medium and then put them in a poor medium or on these gypsum blocks. It does seem that you're asking the question not why potentially they can sporulate, but why do they sporulate under certain conditions? And I don't think that's very well understood. It's just really, nobody's really come up with that answer, I think.
I thought the newest theory was that sporulation doesn't occur because of the stress, that it occurs just as part of the
Normal
natural
growth
cycle. That's what the food science people here are saying, at least.
Yeah.
Oh.
I was talking about bacteria, but you were talking about heat-resistant strains.
Yeah. Well, it doesn't happen during the growth cycle. It happens at the end of the growth cycle for this one.
That's
Yeah.
what they're eating
Yeah.
over food science.
What kind of yeast? Which
He
yeast?
wasn't talking about yeast, he was talking about
Spore
bacteria.
you're talking about.
Oh, don't confuse.
No,
You don't talk about-
this is in food mycology.
Oh yeah, well that would be about yeast. Yeah. Well, I think another thing too is that each yeast has its own particular ways of and reasons for sporulating, and the one that's been mostly studied has been Saccharomyces cerevisiae.
I'm wondering, when you say this is a sexual cycle, I never,
Mm.
this just struck me. Is there any conjugation between the
Yes.
two?
First, this is you get a meiotic division, so that you have this now as in the haploid phase, and then either in the ascus like this, they'll conjugate back together again, or more often than not, this will release these spores, and then they'll join together and conjugate and form a new
So there is,
vegetative phase. Yes, it's
interrelation.
right, and right, and this is very well studied, the genetics of this of the crossing, the conjugation, or what happens after conjugation. Well, I think our time is up. We have lab today, and so section 1, if I hope everybody's still in the same section, we'll go in 10 minutes, let's say, in the middle fermentation room. They've got it set up for a tasting, so just sit down and try not to spill the water or something like that. And tonight, section 2 come in at 7 o'clock at the same place. And then on Thursday, we won't have a regular lecture. At 1 o'clock, section 1 will meet in the Enology building, and at 7 o'clock, section 2 will meet in the Enology building. Okay?